Hello everyone, and welcome back this week. I hope you had a fantastic weekend and are looking forward to an even better week. A couple of housekeeping things. We are in week 12 of our semester. We only have a few more weeks left to go. In fact, after this week, we only have 3 weeks of lecture left, and then the week after that, so 3 weeks of class, and then just 1 week for finals week. So we are really winding down here. With that being said, please keep in mind the final withdrawal date for the college. So for any of your classes, if things just have not worked out the way that you wanted them this semester due to unforeseen circumstances, it's no big deal, but the last day to withdraw is this Friday, April 17th. So if there's some things that maybe just haven't worked out in your favor for reasons out of your control or whatever it may be, the final withdrawal date is on the 17th, which is Friday. This week we will finish up acid, base, water, and electrolyte balance in our notes. We may even get a chance to start on the reproductive chapter. That may happen on Thursday. There's a good chance that that will be the case. And then in lab class this week we're going to do reproductive, and then we are going to have the rat dissection on the next week, and that's what our final lab practical is going to entail. There have been some, it's got to be a slight schedule change. I've already told you about that we're not going to have that exam that's on the 30th, so that fourth exam that's on the 30th, remember to cross that off of your schedule. We're not going to have that over just water and electrolyte balance and reproduction. We're not having that exam. But a little bit of a schedule change for your lab practicals. Currently, your fourth lab practical is scheduled to be given the week before finals week, which is on May 7th, but I am going to have to change the date of your lab practical to April 30th. Now, I'll also send out an email announcement of this, so if you're listening to these lectures a little bit later, that you're on a little bit different schedule, I will send an announcement out on Blackboard, and I'll also discuss it in lab class this week, that your last lab practical over reproductive and the rat dissection will happen on April 30th. So it's still a Thursday. Everything still works the same way that it had before, but our time schedule is a little bit different here. So as opposed to having it on the 7th, we're going to bump it up 1 week and have it that week. So the same day that you're supposed to have your fourth lecture exam, you're going to have a lab practical on that day. Everything else is still going to be pretty much the same. Your final, remember, is comprehensive. It will be in our lab classroom during your normal lab time, and that's really about it. So I can't think of anything else right now. So without further ado, let's go ahead and get into acid and base balance. Now, we've already talked a little bit about acid and base balance in the urinary chapter. We understand that the pH of your blood likes to be between 7.35 and 7.45. Anything lower than that is considered acidosis. Anything higher than that is considered alkosis. The pH of your body fluids can be changed by adding hydrogen ions or adding hydroxy ions or removing them, so it's a constant fluctuation that has to constantly be monitored. Acids and bases may be strong or weak. Whether or not they're strong or weak just depends on how much they're going to dissociate in solution. If it is a strong acid or a strong base, then it will completely dissociate into solution. So what that means is that the ions that make up that strong acid or strong base will be completely broken apart. They won't be one molecular unit any longer. Weak acids or bases, they don't completely dissociate in solution, so that means that some of the molecules, some of the molecular structure of that acid or base is going to still kind of be intact a little bit, as in the case of bicarbonate ions. They liberate few hydrogen ions, and then the change in the pH is not nearly as dramatic as we would have for a strong acid or base. So they have less of an effect on the pH of the solution in which these weaker acids or bases are found. So an example of a weak acid is carbonic acid. Oops, that's not what I wanted. It's carbonic acid. It is a weak acid. In extracellular fluid at normal pH, this is the equilibrium for that reaction, that this is our carbonic acid, which we've been talking about all semester long, from the respiratory system to the digestive system, and even in the urinary system we've talked about this, that when it dissociates, it will break down into a hydrogen ion, which we see here, and a bicarbonate ion. Acidosis is a physiological state that makes your pH lower than 7.35, which I'm going to highlight this, but I'm pretty sure you all are familiar with this. Alkosis is any physiological state that results in the pH being higher than what it normally be, so anything greater than 7.45. And this is exactly the same as what we had at the end of the urinary system. So different kinds of acids that we could talk about are, we have fixed acids. And fixed acids, one of their hallmarks is that they don't actually leave the solution. Once they're produced, they remain in your body fluids until your kidney eliminates them. Some examples of this are sulfuric acid and phosphoric acid. So these are important fixed acids for amino acids, phospholipids, and nucleic acids. So these acids here, they're not going to actually leave out unless they're eliminated by the kidneys, and only what's eliminated is what's considered waste. Anything that's used to build an amino acid or your phospholipids, which just make up your plasma membrane of your cells, your cell membrane, or your nucleic acids that make up your DNA, those are going to stay into circulation. So that's why we call them fixed, because they're just going to hang there. They have a purpose, and they're constantly being used. Anything that's extra, then that's what's going to be eliminated by your kidneys. Second type of acid we have, so the first type was a fixed acid, the second type is an organic acid. Organic acids are going to be the byproducts of aerobic metabolism or cellular respiration. They're metabolized pretty quickly, pretty rapidly, so they don't really get a chance to accumulate. Lactic acid is an example of an organic acid that's produced by anaerobic metabolism, and for these that are produced by anaerobic metabolism, they build up really quickly. And as you remember from ANP1, the way that your body deals with this excess lactic acid buildup is to convert it back into pyruvate through the Cori cycle in your liver. So lactic acid is considered an organic acid, but you also have succinic acid. That's going to be one of those intermediate byproducts of your Krebs cycle. Malic acid, also in the Krebs cycle. Those acids there, malic acid, fumic acid, succinic acid, they don't get a chance to accumulate very quickly because they're going to be converted into different byproducts as we go through the Krebs cycle. So if you'll go back to the Krebs cycle in your book and just kind of make note of some of these acids we're talking about, succinic acid, fumic acid, malic acid, those are organic acids that we're talking about that are produced by aerobic metabolism. And those, as you will watch, they kind of get converted into different things along the Krebs cycle. So that's why they don't get a chance to really build up and cause a huge drastic change in your pH, because they're constantly being converted to different compounds, or what we consider intermediate compounds. Lactic acid, on the other hand, well, that's kind of the byproduct of lactic acid fermentation, where if you don't have enough oxygen available, you will start to build this lactic acid. That's a little bit more problematic, that has a greater ability to affect pH, and it builds up very quickly. For every glucose molecule that goes through this anaerobic metabolism, you're going to make this byproduct of lactic acid. But your body has the Cori cycle, and if you're not familiar with that, you can always read about it in the muscular chapter, which I think is chapter 9 or chapter 8, about how your body deals with that lactic acid. And we have our carbonic acid, which we've talked about. Carbonic acid is a volatile acid that can actually leave solution and enter the atmosphere. Since it can leave solution and enter the atmosphere, you're probably already thinking respiratory system. So while at your lungs, the carbonic acid breaks down into carbon dioxide and water. And remember, that was one of the ways that we could get carbon dioxide back to the lungs, was to convert it into carbonic acid when we had that the chloride shift, where we brought in chloride for this carbonic acid, and then that carbonic acid, when it was in the plasma, would go back into the lungs, and then we have that shift again where we converted back into carbon dioxide and then exhaled out. So that carbon dioxide, which has been carried by this carbonic acid, then diffuses across the alveolar wall. So what you're probably noticing here is that in this chapter, we're kind of touching on a lot of the different organ systems that we've talked about, not only here in ANP2, but also an A&P1. So you're going to repeatedly and quite frequently hear me refer back to things that we've done in A&P1 or things that we've done at the beginning of the semester, because all of the things that we're talking about, for the most part, we have discussed before. So it's a really good refresher before your final. So carbon dioxide and pH are intimately tied to one another. That we understand and we know from the respiratory system and also from our discussion of respiratory compensation in the urinary system at the very end of those notes. Most carbon dioxide that's in your solution converts to carbonic acid. Most of that carbonic acid is going to dissociate. The partial pressure of carbon dioxide is very important for affecting the pH in your body tissues. Partial pressure of carbon dioxide and pH are inversely related. So as one goes up, the other one goes down. So as the partial pressure of carbon dioxide increases, that means our respirations are very low. We're holding on to this carbon dioxide. We're not exhaling as quickly as we normally would. So we have a higher concentration of carbon dioxide, then our pH is going to be lower. So we're going to have more of an acidic pH. Conversely, if we're hyperventilating and we are blowing off that CO2 very quickly and there's a lower partial pressure of carbon dioxide, then the pH is going to raise higher and be more alkaline. And this is not a new concept. This is just revisiting an older concept about respiratory compensation, the role that pH and carbon dioxide, the relationship they have with one another. It's just giving you a different perspective on that same concept. So when your CO2 levels rise, hydrogen and bicarbonate ions are released, so your pH goes down. So when you're holding on, so when you have a high concentration of carbon dioxide, your pH is going to go down. Now what's happening in your lungs? At your alveoli, this carbon dioxide is going to fuse into the atmosphere. Hydrogen and bicarbonate ions in the capillaries are going to drop. And as a result, the pH of the blood is going to rise. So here's a good picture of what we're showing here, that when we have our normal partial pressure of carbon dioxide between 40 and 45 millimeters of mercury, we have reached homeostasis, so we're nice and balanced there. pH is what it needs to be. If the partial pressure of carbon dioxide rises, then what's going to happen is that we're going to have this carbonic acid dissociate into hydrogen ions and to these buffers, these CO3. So when carbon dioxide levels rise, more carbonic acid forms, additional hydrogen ions and bicarbonate ions are released, and as a result, the pH goes down. So here's that inverse relationship. As partial pressure of carbon dioxide increases, the pH is going to decrease. And remember, things that can increase the partial pressure of carbon dioxide would be hypoventilation, where you're not exhaling as frequently as you normally would. So anything that's above 45 millimeters of mercury would be considered hypoventilation or considered a higher level of carbon dioxide. Conversely, and actually this picture here, we're looking at pH. So if your partial pressure of carbon dioxide falls, the reaction runs in reverse, and carbonic acid dissociate into carbon dioxide and water. This removes hydrogen ions from solution and increases the pH. So when your partial pressure of carbon dioxide goes down, when you're exhaling very rapidly, with hyperventilation, you're blowing off all that carbon dioxide so quickly that it doesn't really get a chance to accumulate, so that we're not in that 35 to 45 millimeters of mercury range for the pressure of carbon dioxide. So what's happening there is that this the hydrogen ions here, so the hydrogen ions from the solution are going to be bound with the bicarbonate ions, and this bicarbonate or this carbonic acid will then break down into water and carbon dioxide, which you're exhaling off. So when I say you're blowing off those hydrogen ions, well, you're getting rid of the CO2, and then that hydrogen that was part of the carbonic acid, well, that formed with the water. So as a result, when you're hyperventilating, when your pH when the partial pressure carbon dioxide goes down, then your pH is going to rise and be more alkaline. And remember, this is the same concept that we talked about at the very end of the urinary system with acid and base balance, just a little different view or presentation of that same material. So in order to maintain your acid and base balance, your body balances the loss and gain of those hydrogen ions as well as the loss and gain of those bicarbonate ions, which you're probably very familiar with. So HCO3 minus are those bicarbonate ions, and H plus are those hydrogen ions. So it's a constant ballet of these hydrogen and bicarbonate ion balances that allows us to maintain our pH. So hydrogen ions, where do we get them from? Well, we get hydrogen ions from our diet through our digestive tract, and then we also get hydrogen ions through metabolic activities. How do we get rid of hydrogen ions? Well, we can eliminate hydrogen ions in our urine, and we can also eliminate them in our lungs. And you probably are pretty familiar with how you can eliminate them from your urine, but this is how you can get rid of them from your lungs. Remember this little dandy formula we had here? With this formula here, we had those hydrogen ions hooking up with that buffer of bicarbonate, makes that carbonic acid. When your carbon dioxide levels fall, and then here is that water. That hydrogen ion, that same hydrogen ion here is here in the water, and then you have your carbon dioxide. So we can get them eliminated at the kidneys. We have to neutralize the concentration of our hydrogen ions to avoid tissue damage, and this we talked about in digestive, that for your parietal cells, even though they make hydrochloric acid, they don't make it directly in the cell because those hydrogen ions can be very damaging to it. So instead, what they have is the chloride tie, or the alkali tie, that is going to make the hydrogen and the chloride in different parts of the cell or different processes of the cell and then shift them into the inside. So acids produced in normal metabolic activity are neutralized by buffers such as bicarbonate in your body fluids. So it's a buffer system. There's a couple of buffer systems that we're going to talk about. And anytime we talk about a buffer, it consists of the combination of a weak acid and an anion released by its dissociation. So HCO3 minus is considered an anion. Anything that has a negative charge is considered an anion. In the terms of physiology, the anion in question here that we're talking about will act as a weak base. So in solution, molecules of a weak acid exist in equilibrium with its dissociated products, and this is what allows it to be balanced out so that it's not an acid, it's not a base, it's neutral. So we have three major buffer systems: protein, carbonic acid, and the phosphate buffer system. The carbonic acid bicarbonate buffer system, you're very familiar with that. It's very important for your extracellular fluid to maintain that the appropriate pH. Protein buffers we haven't really talked about much, but they help to regulate the pH in your extracellular fluid as well as on the inside. So protein buffers, as the name would suggest, are proteins or amino acids that will pick up excess hydrogen ions. And with protein buffer systems, they quite frequently will interact with other buffer systems, such as the carbonic acid, bicarbonate, or phosphate, particularly phosphate buffer systems. And then the final buffer system that we'll talk about is the phosphate buffer system, and it mainly buffers the pH of your intracellular fluid as well as your urine. So I'm going to highlight the names of these buffer systems, and I would like for you to remember where they help to regulate the pH. All right, so protein buffer solutions. Protein buffer solutions, as you probably have already guessed, are completely dependent upon amino acids. They're going to make their changes or respond to pH changes, and they will accept or release hydrogen ions. If your pH rises, the carboxyl group of the amino acids, for each amino acid, there are various parts to them. You have an R group, which there are 20 different R groups. That changes for those 20 different amino acids. You have a carboxyl group on there as well, and you have an amino group. So it's the carboxyl group, that portion of the protein that we're talking about now. So if your pH goes higher, the carboxyl group of amino acid disassociates. So that COOH is going to disassociate, and it's going to act as a weak acid, and it will release a hydrogen ion. So the carboxyl group, and I'm just going to draw this or write this out here. is COOH. Now this is one of those classes of functional groups that you probably learn in intro biology. So you had COOH, that's your carboxyl group. So if your pH gets too high, it's higher than 7.45, then we need to add more hydrogen ions. So if we dissociate this carboxyl group, we can have CO2, carbon dioxide, and we can have a hydrogen ion. So there's that release, and I'm sorry, my handwriting is so horrible with the mouse, that hydrogen ion. And then this carboxyl group becomes a carboxylate ion. So this is what that is, a carboxylate ion. The carboxyl group of most amino acids have already given up their hydrogen ions, so let's look at what happens when the pH drops. So when your pH drops here, the carboxylate ion and the amino group act as a weak base, and they're going to pick up hydrogen ions. So then you have that regular carboxyl group form and an amino group. So the equation that we drew before, it just goes in the opposite direction. So this carboxylate ion now can pick up excess hydrogen ions. So opposite charges attract. It can now act as an acceptor. This carboxylate ion can act as an acceptor, and this is where we have our carboxyl group, again, to make, that can be used for our amino acid. So not only can we use carboxyl ion, sorry, not ion, but this carboxyl group as a buffer, but we can also use this carboxyl group to make amino acids. Hemoglobin buffering system. Here we've talked about this before with the carbonic acid. Carbon dioxide diffuses across the red blood cell membrane, and it does so just as a result of diffusion, gas law diffusion. The gas moves from an area of high pressure to low pressure, high concentration to low concentration. As that carbonic acid dissociates, we make that bicarbonate ion, and when we switch out the bicarbonate ion that we made on the inside of the red blood cell, we wanted to put it out into the plasma. So we bring in a chloride ion for that bicarbonate ion, and that is that chloride shift that we talked about before. Hydrogen ions can be buffered by the hemoglobin molecule. So if you go back, way back to one of the very first chapters we did, we showed how that when we took that carbonic acid and broke it down into bicarbonate and a hydrogen ion, that hydrogen ion could bind directly to hemoglobin. So this concept is not, for you, believe it or not, new at all, because this is something we talked about very early in the semester. And here's the exact same picture that we looked at very early in the semester. So here we go, that we had our carbon dioxide. Some of it can, remember we talked about those various methods by which we can get carbon dioxide back to the lungs. So we're in the systemic system, we're not in the lungs yet. So the leftover CO2 that is a byproduct of metabolism, some of it can ride in the plasma to get back to the lungs. Some of it is going to actually bind to hemoglobin, and then we had some of it, on the other hand, that would actually combine to make this carbonic acid. And then this carbonic acid would break apart into the bicarbonate ion here and the hydrogen ion, and then this hydrogen ion can bind to hemoglobin. This bicarbonate ion then will be put out into the plasma and will bring in chloride in its place. And then once we get back to the lungs, the opposite happens. So let's just work with the chloride shift again. That bicarbonate ion is going to come back from the plasma, reassociate itself with that hydrogen ion from the hemoglobin, and then we have this carbonic acid. Carbonic acid can be broken down into water and CO2. This CO2 is exhaled out. Now we have talked about how this process here can alter the change of pH. The more carbon dioxide that you hold on to, that you're hypoventilating, so the higher your carbon dioxide concentration, the lower your pH. The lower your carbon dioxide concentration, hyperventilating, the higher your pH. So you can see a picture of how this relates to what we just talked about in how the respiratory system and carbon dioxide have a role in pH. So there we have that process happening there. So this, as I said before, this is an image and this is a concept that you've talked about already. So just more reviewing. So the carbonic acid bicarbonate buffer system here relies on carbon dioxide. Most of your cells are always going to constantly generate carbon dioxide because it's a byproduct of metabolism. Most carbon dioxide is converted into carbonic acid, which disassociates into a hydrogen ion and a bicarbonate ion, which we saw on the previous slide. This is formed by carbonic acid and its association products, like we saw, the process happening on the previous slide. And it keeps changes in pH by organic acid and fixed acids in your extracellular fluid. So it prevents those major changes from those different types of acids between organic acids and fixed acids, because we have these bicarbonate ions that are made. And then our final buffer system is our phosphate buffer system. Consists of an anion. Works like a carbonic acid buffer system, and it's most important in maintaining the pH of your intracellular fluid. So what it means by saying it works like the carbonic acid bicarbonate buffer system is that we can get this PO4 minus out, and then we can have this hydrogen ions that they'll disassociate in the same way that carbonic acid disassociates into a hydrogen ion and a bicarbonate ion. Now, for those three buffer systems we talked about, and we talked about the bicarbonate buffer system, we talked about hemoglobin acting as a buffer because it can accept anything that can accept excess hydrogen ions as a buffer for the most part. We talked about protein buffer systems that can, where the carboxyl group can act as a buffer and be converted into a buffer if there are changes in the pH and the pH gets too high. And we talked about the phosphate buffer system. Now, some limitations to that. They can only give us a temporary solution to the acid-base imbalance. They're not really going to eliminate hydrogen ions in the same way the urinary system will eliminate hydrogen ions. They really just tie them up, so they occupy them, so they're not just floating around by themselves. But your supply of buffer molecules is limited. So remember, we have to make these molecules as a result of various physiological processes, whether it be the breakdown of the amino acid into its carboxyl group, so tearing that carboxyl group off, that's going to be limited by the amount of free amino acids that you have. The hemoglobin, that's going to be how much hemoglobin is available to act as a buffer, is going to obviously be tied up and related to oxygen and carbon dioxide concentrations. The same thing with your phosphate buffer systems, that there is a metabolic process to make them as well as to break them down. So there are a couple of limitations to buffers, and one of which is simply that the supply of these buffers is going to be limited, that you don't have just a steady, unlimited supply, and there are some energetic costs with building these molecules and breaking them down. So next discussion we'll have is overcompensation. And remember, we already talked about compensation a little bit at the tail end of the urinary system when we did that acid and base balance. So respiratory compensation is going to occur whenever the pH moves outside of your normal limits. So let's highlight that one. And what respiratory compensation directly affects is your carbonic acid bicarbonate buffer system. So this one here that I'm going to draw for you, this is the buffer system that was HCO3 minus. This is the buffer system that we're referring to, and just to refresh your memory more, this one here. So increasing or decreasing your rate of respiration is going to alter pH by lowering or raising your partial pressure of carbon dioxide. This is probably the third time that we've made this statement, and we've talked about this in the respiratory system in our acid and base balance discussion at the end of the urinary system. Even in this chapter today, we've talked about this. Remember, partial pressure of carbon dioxide and your pH are inversely related to one another. So what does that mean again? If one goes up, the other one goes down. So if the partial pressure of carbon dioxide rises, as you've probably already surmised, pH is going to fall. Addition of carbon dioxide drives the buffer system to the right. So what we mean by that is that if your pH is falling down, that means it's much too acidic, then we need to tie up some of those excess hydrogen ions. So we'll do that with the bicarbonate ions. So that's what we mean by driving it to the right. Look at the formula we have here. Let's grab a pencil out. Minus, that's that carbonic acid. And that's that bicarbonate. And conversely, if the pH falls, or the partial pressure of carbon dioxide falls, then the pH is going to rise. Removal of the carbon dioxide drives the buffer system to the left. So we are making more, untying, I should say, more of these hydrogen ions. Here's a pretty picture of all of this stuff. So at our normal concentration or normal partial pressure of carbon dioxide, everything is all good. There isn't a problem with it. If the arterial partial pressure of carbon dioxide increases, what we call hypocapnia, you have a higher concentration of that, then things that are going to happen is that the decrease in the partial pressure of carbon dioxide is going to decrease the pH, and you also have the stimulation of the arterial chemoreceptors that we're going to stimulate the respiratory muscles. By having that decrease in pH, it also stimulates respiratory muscles as the chemoreceptors at the medulla oblongata, all in an attempt to increase your respiratory rate, so to increase the amount of respirations, the time that you're exhaling out, to get rid of that CO2. So by getting rid of that CO2, then we're able to bring our pH back into normal parameters, which in this case, bring it up. And then conversely, on the other end of it, if our partial pressure of carbon dioxide is decreased, hypocapnia, it's very low, then it's going to increase your pH. That will inhibit your chemoreceptors, your arterial chemoreceptors. That will inhibit respiratory muscles. The pH change that's happened that's too high now reducing stimulation of those chemoreceptors, and then decreased respiratory rate, which decreases the elimination of carbon dioxide, which brings our pH back up to normal and brings our partial pressure of carbon dioxide back to normal as well. All right, so for renal compensation, this last little bit here, we will finish this up on Wednesday. So we'll talk about renal compensation again, because we talked about it again, end of the urinary system, and finish out this chapter and then start on reproductive. Have a wonderful day.